We're going to talk about the muscular system. Muscular system is our next big system. We talked about the bones, which serves as the framework, number one, to provide us upright and for protection. And now we want to move, okay? So one of the things we want to look at is movement, okay? Maintaining posture, stability, and heat production. Movement, maintaining posture, stability, and heat production. Let's see, slide. Comparison of muscle types, universal characteristics. Number one, all muscles are excitable, okay? They respond to a signal. Muscle cells only do one thing. They contract. They contract. They contract in the direction that the fibers are associated. And it doesn't matter where you stimulate the muscle cell at. You can stimulate it in the, usually the nerve end plate that we'll talk about is in the middle of the muscle cell, but you could stimulate it some other place and the cell still contracts. It doesn't do anything other than that, okay? So it's got to be able to be excitable and respond to a signal, okay? It's got to be able to shorten. That's all a muscle cell does is shorten. It shortens and relaxes. It shortens and relaxes. It's got to be able to be stretched, okay? And finally, it's got to be able to return to its resting length. So a muscle is excitable, it's able to contract, it's able to extend, and able to return back to its initial length. All a muscle cell does is contract. A nerve impulse comes out, comes down from the central nervous system, tells this muscle cell to contract. If you get enough muscle cells to contract, you're going to move something, okay? Skeletal muscle, there's three types of muscles. We remember that from our tissue thing. There's skeletal muscle, there's cardiac muscle, and there's smooth muscle. Skeletal muscles are long, they're unbranched, and they're cylindrical. They don't need to be branched. We've got a whole bunch of them. We've got a whole bunch of them. We don't need them all to fire off in the exact sequence. If I am doing something with my biceps femoris and I fire 1,000 muscle fibers off at time 0 and another 100 fire off 1 millisecond later, I'm still going to contract my biceps. Whereas in the heart, like I explained to you, we want to start that squeeze in the ventricles to the apex and squeeze upward because the valves that go out to the lungs and to the aorta are superior to the apex of the heart. So we need to push that blood up and we need to push it up in a sequential fashion. The skeletal muscle cells are multinucleated. The bulk of the cell is composed of myofibrils, okay? Those are myofilaments in that regular pattern. Remember when we looked at muscle cells, especially skeletal muscle, they have that nice striations on them, okay? They have an extensive tubular system. Instead of calling it endoplasmic reticulum, in a muscle, we call it sarcoplasmic reticulum. And we have sarcoplasmic reticulum, which is endoplasmic reticulum, but now it's in a muscle cell. It is primarily for storage of calcium. We need calcium for a muscle cell to contract. And in that complex, we have T-tubules. The skeletal muscles are striated. They are voluntary. You and I control them at this very minute. And most are attached to bones. That's how we move, okay? That's how we move. That is how we're keeping ourselves upright at the moment, unless you're laying down to listen to this lecture or reclining, okay? But they're multinucleated. They have striations, okay? And they have this extensive tubular system. This is skeletal muscle right here. It has these very nice striations here. We looked at that before. We will learn what all those striations are and what the names of that are. And we'll do that in just a little bit, okay? That is skeletal muscle, multinucleated. Fibers are cylindrical, okay? They are not connected. And there's a sarcoplasmic reticulum there. Cardiac muscle, cells are branching and cylindrical. They're branching because you want one cell to connect to the next cell to connect to the next cell. So that when we squeeze from the apex to squeeze blood out, of the heart, either on the right side to the lungs or the left side of the aorta. We want to do it sequentially. They are uninucleated. They have less well-developed sarcoplasmic reticulum, fewer myofibrils, more mitochondria. So this is in the muscle, they call things different words. I'd actually never saw this word before in my life until I got these slides from Dr. Oler. But they call it myochondria or mitochondria. They also have lipid and glycogen deposits. They are striated with intercalated disc. Okay, those discs connect one muscle cell to the other muscle cell. They are self-stimulating. You know, we have an SA node and AV node. You'll learn about that. That is, that will be chapter 18 when you take Anatomy and Physiology 2, either next semester or the following semester. Remember, you don't want to wait longer than one semester, okay, because you lose knowledge exponentially unless it's reinforced. It is self-stimulating and involuntary, and it's only in the heart. We don't have that muscle in any other place except the heart. And, you know, this is our picture of skeletal muscle. These are the intercalated disc. These are one muscle fiber attaching to another muscle fiber, and that's cardiac muscle. And finally, the last type of muscle is smooth muscle. It's slender, spindle-shaped, uninucleated. So based on the three types of muscle, if you get a question that says this type of muscle is multinucleated, since we said that cardiac muscle is uninucleated and smooth muscle is uninucleated, one nucleus, then if it's multinucleated, it had to be skeletal muscle or voluntary muscle. Limited number in gap junctions in some, skimpy sarcoplasmic reticulum, myofilaments are less regularly arranged, they're not striated. This is involuntary muscle. This is a muscle that works on your smaller blood vessels to open up capillary beds and close capillary beds. It's also the muscle that works in your respiratory system to close off non-functional alveoli. And lastly, it's the muscle in the GI system. There's two layers of it, a longitudinal layer and an intercircular layer, okay, that are responsible for mixing up nutrients that we have eaten and for moving the nutrients down. The mixing up nutrients is called segmentation. You don't have to remember this, but the moving the nutrients through is peristalsis, okay? Peristalsis is responsible for that. And so it is normally found around hollow, viscous internal organs, okay? And then that's smooth muscle there, okay? That's smooth muscle there, uninucleated. Structural organization, okay? From nerves and muscles, we're going to have three layers. We're going to have three layers, okay? This is an individual muscle cell, okay? This is an individual muscle cell that is composed of a number of fibers. The biggest bulk of skeletal muscle is these myofibrils, myofilaments, because this is what's going to allow us to have this muscle to contract, okay? Well, around this individual muscle fiber here, we have a cover, and it's called an endomysium. An endomysium. So an endomysium is around this individual muscle fiber. If we have a bunch of muscle fibers, okay, together, then the next coating is called a paramecium. So endomysium, endo, inside, okay? Endomysium is the covering of the individual muscle fibers, okay, muscle cell. Paramecium is the covering around a group of muscle cells. We usually call that a fascicle. And then when we join all the fascicles together into the muscle, okay, we have an epimysium, okay? And so you're going to learn this because we'll have an epineurium and an endoneurium when we talk about the nerve, okay? I was watching, there is a lady that cooks on Facebook. She runs a bed and breakfast, and I like to watch her cook. And she was making a ham this afternoon. Well, she wasn't making a ham. I was watching the video about her making a ham. and she took out the ham, and you could actually see the fascicles. Okay, you could see the fascicles here, the individual portions of the muscle. Okay, so this is an individual muscle fiber or muscle cell made up of a bunch of muscle fibers. It has an endomysium around it. When we have a bunch of muscle fibers congregate together into a fascicle, we call that a paramecium, the covering outside covering. And then when we have the entire muscle here, the covering on the outside is the epimecium, okay? So those are the coverings. So the endomysium surrounds each muscle cell. A fascicle is a bunch of muscle cells. Paramecium surrounds the fascicle, and the epimecium surrounds a bunch of fascicles. So the muscle, we have a covering around the muscle itself. Fascicular arrangement, okay? We can have it as a fusiform, which is thick in the muscle, thick in the middle, and tapers on both ends. Biceps femoris, okay? Our good old biceps muscle, okay? Excuse me, biceps femoris is in the leg, okay? The biceps in the arm is a fusiform muscle. All the fibers are in the same direction. They run parallel, okay? Fibers run parallel, fusiform. And that's an example of a fusiform muscle. A convergent muscle is thick attachments on one end, and then it tapers to the other end, usually to a tendon. This would be the pectoralis major muscle. Pectoralis major muscle, thick on one side, tapers to an end on the other end, which is usually into a tendon, okay? Remember, ligaments attach bone to bone, tendons attach bone or muscle to bone, okay? So we have a convergent, okay? Then we have pinnate, okay? We have three different types of, well, we have more than three different types of pinnate. You have a central tendon, in a muscle, okay? You have a central tendon in a muscle. If we only have one muscle group attached to the central tendon, and that they describe this as feathery looking, okay? It's a unipinnate. Uni means one, okay? See, pinnate means feather. If we have two, so that's extensor digitorum longus. I'd never ask you an example of that, okay? Bipinnate is the rectus femoris. This is one of your quadriceps muscles in your leg, rectus femoris. Here's a central tendon. We have two of these feathery-like structures. The fibers are running in the sort of transverse diagonal direction. They're going to attach to the central tendon. And then we have a multipinnate. This is in the deltoid. Where we have three, or we have more than two of these feather-like muscle structures that are going to attach to the central tendon. And so that's pinnate, feathery-like. And then circular are the fibers that run in a circular fashion. We have two, basically. We have more than two, but we have the orbicularis oris. Oris means mouth, so it's the round fibers around the mouth, okay? Then we have the orbicularis oculi, which are round fibers around the eye. And so they run it in a round fashion. One of the local plastic surgeons, in the Jackson, many years ago, I think this fellow's dead now, but many years ago, one of the trumpet players for the Jacksonville Symphony Orchestra ruptured his orbicularis oris, and obviously if you're a trumpet player with a ruptured orbicularis oris, you'd have a problem. So he repaired that for him. Muscle attachments. We can have direct muscle attachment to the direct muscle epimecium to the periosteum of the bone. This is the temporalis, okay? We have the epimecium. Remember, we have endomysium, paramecium, and epimecium. Epimecium is the outer covering of the entire muscle. This epimecium is attached to the periosteum of the bone, okay? This epimecium is attached to the periosteum of the bone and then comes together, converges into a tendon, okay? We have indirect, okay? We have indirect. This is the master here. This tendons attach muscles to bone, actually penetrating the bone. So we have the master muscle responsible for closing the jaw that is attached, obviously, to the angle of mandible, but actually sends this tendon up to the zygoma, okay, zygomatic arch to which it's attached. Okay, that's another type of attachment. And then the last type of attachment is when we have muscle attached to thickened connective tissue. This is called the aponeurosis of the skull. Okay, when you do an incision in the skull, if you did an incision up here, you go through the skin, subcutaneous tissue, you would have this white aponeurosis that you would incise. It's not very thick, a couple of, maybe 3 millimeters or something like that. And then you would see bone, okay? Well, the frontalis muscle and the occipital muscle, okay, attach this aponeurosis. That's another type of attachment. Now we're going to look at the microscopic anatomy, okay? Those nice striations that we talked about, we're going to look at the microscopic anatomy of that. Sarcoplasm, okay, is similar to cytoplasm, but contains glycogen stores, okay? We have a sarcolemma, okay? Remember, in a cell, just when we're talking about cytology, we have a cell membrane. In a muscle cell, we have a sarcolemma. It's the same thing. It's the cell membrane of a muscle cell. Instead of having cytoplasm in a muscle cell, we call it sarcoplasm. Okay, but these are not quite unique, but two places that glycogen is stored is in the liver and in the muscles. If you think about it, it needs to be in the muscles for our old-time fight or flight reflex, okay? When we need to get up and get going in a hurry, okay, we need to be able to have nutrients for the muscle, okay? Well, we actually have glycogen, we have glucagon stores, we have glycogen in the muscle, and the glucagon is going to be broken down if we need immediate energy. Okay, we have some glucose and some ATP, but this is our immediate energy source because all glycogen is a long string of monosaccharides. Okay, we have mitochondria, which is the source of ATP. Remember, we're going to need a lot of mitochondria. And we have myoglobin, which is the hemoglobin associated with the muscle cell. Okay. We are now going to look at the striations, the particular striations associated with what we saw in the picture here, the A band and I band. Okay, this is the structure of an individual myofibril. Okay, you're going to need to learn these names. Okay, you're going to need to learn these names and what they represent. Notice that we have an I band, A band, I band, A band, I band, A band, I band, A band. They continuously repeat, okay? The I band has a Z disc. We're going to go over what this means in a minute. They have a Z disc, and the A band has an H zone and an M line, okay? Well, what do these represent? They are two proteins that were responsible for muscle contraction. They are actin and myosin. Actin is the thinner of the proteins represented by the blue line here. Okay, myosin is the thicker of the proteins represented by the red line. In this I band area, we only have actin. We only have this blue protein. In the A band area, in some parts we have myosin and actin, but some parts we only have myosin. But the A band represents where there is myosin. The I band represents where there is only actin. So this red stuff right here is myosin. The blue stuff right here is actin. We're going to look at it in some greater detail, okay? So when we see these striations up here, this is what we're looking at. And the individual functional unit of a sarcomere, which is the functional unit of a muscle cell, goes from one Z disc to the other Z disc. Notice that actin doesn't completely doesn't completely go all the way across here in the A band. There is a portion called the H zone where you only have myosin. There is a portion called the H zone where you only have myosin. And you have an attachment in here, which is called the M line. You really have two attachments. One is this Z disc, and one is the M line. And we'll talk about those in a little bit greater detail. So you'll need, obviously it's going to be easier for you since you have open book, open notes, but this is the structure. The sarcomere goes from a Z disc to a Z disc. We have two proteins in here. The thinner one is actin, the thicker one is myosin. And what they're going to do is literally slide over the top of one another, and that's going to cause the contraction. Okay, so now we're looking at this in a little bit greater detail. Okay, this is the Z disc. Okay, this is the Z disc. The I band would be from, if we would start here at the end of the myosin and we go over here to the other side where the myosin started, that would be our I band. That, notice we only have actin in that area. We only have actin in that area. So we have a Z disc here. This is where we have attachments of the myosin to the Z disc. Okay, we got attachments of the myosin to the Z disc, and the actin attaches here. So this is a point of, we have this elastic fiber called tentin that attaches the myosin to the Z disc, but we also have the actin attached to the Z disc because we need a firm foundation when these are going to contract and slide over the top of each other. So this is a Z disc. This would be an I band, okay, would have been the end or start of the myosin here to the end or start of the myosin over there. That would be our clearest of bands because it only contains actin, okay. Our A band starts when we have myosin, so it goes from here to here. That is our A band. That is our A band. Oh, here it has elastic fibers 10. The thick myosin filament. Notice in this particular area, okay, we have our H zone. We have our H zone right here, okay, where we only have myosin here, okay, we only have myosin here. This is our H zone where we only have myosin. We don't have any actin because the actin is ended there. And then this is our M line, which is also an attachment for the myosin, okay? It's also an attachment for the myosin. So this is a schematic of this diagram up here about what this looks like because of a individual sarcomere. Sarcomere is the functional unit of a muscle, goes from one Z disc to the other Z disc. So in here, it go from here, go from there to there. That would be a sarcomere, okay? Myofibrils contain the actin and myosin. Actin is a thin filament. It contains, okay, tropomyosin and troponin. Myosin is a thick filament. Tentin is the elastic fibers, okay, that secure the myosin to the Z disc. And I want to show you this picture here. We'll look at this just a second. This is actin here, okay? Imagine actin being a bunch of pearls, okay? Imagine actin being a bunch of pearls. So you put all these pearls in a linear fashion and you make you two strands of these beads, okay? And you put them together, okay? You make it two strands of these beads and you put them together. So we got two strands of these beads up here putting together, but we didn't run a string through them, okay? We didn't run any kind of thing through them to keep, whether you run a necklace, a chain, or whatever to keep them together, okay? So there is some attraction to these actin subunits, but we also have tropomyosin, okay? So tropomyosin, as you can see, spirals on the outside of these two strands of these actin subunits. So tropomyosin is there to stabilize and add strength to the actin actin molecule, okay? Tropomyosin also if it's in its normal position, is covering the active sites, okay, covering the active sites, which are going to bind with the myosin heads, okay? These myosin heads are going to bind with active sites on the actin, okay? But when the tropomyosin is in a non-stimulated position, it is actually laying over the top of the active sites on the actin. In order to reinforce that, we have this other molecule called troponin, okay? And you can think of troponin as a U-bolt or something like that, or a pop-up thing that's going to swivel out, okay? Or a pop-up thing that's going to swivel out. And tropomyosin, troponin has ATP attached to it, okay? It is attached to the actin, and when a calcium attaches to it, it's going to swing off. It's going to undergo don't say swing off. It's going to undergo a configurational change. And when it undergoes that configurational change, imagine it sliding off of the tropomyosin here. So this little yellow thing is the troponin, okay? It's going to slide off of here, and guess what? Then that tropomyosin is going to unbind over the active sites. So now the active sites are going to be exposed. So we can now have one of the myoglobulin heads attach to the active site of the actin, okay, because the tropomyosin is now slid off of the head. And you can imagine once the head attaches, okay, then it's going to undergo some type of stroke mechanism. It's called a sliding theory. And it's going to contract, and when it contracts, it's going to cause the myosin does not move. It's going to cause the actin to slide inwards to compress the space here, because that's the reason that we don't have actin here. And we're also going to compress the I-band out there, that space there, and we'll see that in an electron microgram. And we'll talk about that a little bit. But we had a picture here, and I want to go ahead and introduce that to you. And this is one of the things that, there again, this is one of the things I never understood in medical school. Obviously, when I got out into general surgery, residency, and plastic surgery, we never had to learn this again. But when I started teaching it again, I finally talking about, well, this could be duct tape. If you think about it, this could be, the tropomyosin could be duct tape around the outside of this actin necklace, okay? And and the troponin is a U-bolt or some kind of thing that undergoes a configurational change. Actin and myosin, okay, and then we have tentin, which is elastic filament that secures the myosin to the Z disc. And we have the A-band where there is myosin, okay, and the I-band areas where there's actin but no myosin. We've gone over that. The H zone is where there is no actin. So it is this place right here, the H zone right here. This is where there is no actin. Notice the actin ends right here. The actin ends right here. The Z disc is composed of connexin, which anchors the thin filaments and connects the myofibrils across the muscle cell width. So the Z disc, okay, is going to have the elastic fibers of the myosin. It's also going to have the actin filaments working with connexin to attach here, okay? And so they're attached at this point, and these fibers are attached at this point. There's one big continuous attachment here. And so this is actually an electron micrograph, okay? This is an electron micrograph. So this is a Z disc. This is a Z disc, okay? This is our I-band starting here, and it would end up over here. It would be an I-band starting here and end up over here. This would be our A-band, okay? This would be our A-band. A-band from there, A-band from there. This is our H zone, okay? Remember, the H zone does not contain actin. Right here in the A-band, there's actin and myosin. So in this darker part, there is actin and myosin, but in this part, there's only myosin. In this part, there's only actin. And then we have our M zone here, okay? We have our M line, which is where the myosin is actually going to attach in here for strength and support, okay? So the myosin is actually going to attach and be continuous through that area. Regions of myofibril, a sarcomere goes from Z disc to Z disc. This is the functional unit of a myofibril, contains actins on both outer edges with myosin in between, okay? It may take you 2 or 3 or 4 times looking at this. Obviously, the interactive physiology exercises, you're going to look at this. The dynamic study module that I talked to you about in Pearson, you're going to look at this. So that's why I took away the reading quiz and the homework quiz and added the dynamic study modules, so it would help you learn about the sliding filament theory. We also have sarcoplasmic reticulum. Remember, we talked about it. This is sarcoplasmic reticulum here, okay? Sarcoplasmic reticulum here, running parallel to the muscle fibers, okay? Where it has a collection area that is called a cistern, okay? In the old days, if you went to visit your grandparents or maybe you went out on the farm, they had cisterns to collect water, okay? This is a collecting area. Sarcoplasmic reticulum in the muscle cell is the place where calcium is stored. Sarcoplasmic reticulum in the muscle cell is the place where calcium is stored. Calcium is required for muscle contraction for the muscle to contract. When we looked at that troponin, okay, when we looked at that troponin, which is called my U-boat, okay, it's undergoing a configurational change. It's already primed, okay. What it needs, it has an attachment to the actin, it has an attachment for ATP, but the other attachment is calcium, okay. So when a signal comes down to a muscle, that's where one of the places the calcium is going to attach to that troponin receptor to cause that troponin to undergo a configurational change. And when that happens, then the binding sites on actin are going to be exposed because the tropomyosin is going to move off of the binding sites. So we have sarcoplasmic reticulum. They're going to collect in these 2 areas, and they're called cisterns. The cisterns are going perpendicular to the parallel muscle fibers. So this is sarcoplasmic reticulum going parallel. And the two collecting areas, these are, okay, going to pouch out, okay, and become fuller, and these are cisterns. Well, between that, this area right here is a T-tubule, okay? The cisterns are for collection of calcium in the sarcoplasmic reticulum. The T-tubules are actually going to go down into the cytoplasm of the fibrils, of the myofibrils, down into the muscle cells and distribute that calcium, okay? And so when we have 2 cisterns and a T-tubule, the T-tubule is the white thing here. 2 cisterns are the collecting chambers right here. When we have 2 cisterns and a T-tubule, that is called a triad. That's called a triad. And so that's a little bit of anatomy. Ultrastructure, okay. Sarcoplasmic reticulum cisterns, interconnecting tubules that surround the myofibril area where the sarcoplasmic reticulum runs perpendicular at the AI junction. The AI junction. The AI junction, remember in the I, we have actin. When we start into the A, we have myosin. So that is the junction where the myosin first starts. This is where these collecting systems are at, okay? Area where sarcoplasmic reticulum runs perpendicular at the AI junction to create a channel. So now we have these interconnecting things, but we have these channels at the AI junction with this T-tubule. A tubule that penetrates in the cell's interior lumen is continuous with the extracellular space. So we need to have calcium coming in, but we need to have calcium going down into the cell. That's what our T-tubule does. And triad, like I say, is just 3 things put together. And here, once again, this is our complex here. This is the troponin, okay, holding the tropomyosin in place. This is the tropomyosin. So these are our beads here. These are actin beads or actin subunits. made into a double and made into a pearl necklace, okay, with 2 strands of pearls side by side in a helix. Well, we need duct tape or wire, or whatever you want to call it, tropomyosin to go along the outside on both sides to bring strength and configuration to this molecule, okay? But this molecule also has active binding sites for those myosin heads, okay? We want those things exposed when we're ready for the muscle to contract, but otherwise we want them covered up because we don't want the myosin attaching to the actin if it's not time for a contraction, or if it's not a concerted effort for contraction. So this is my troponin. That's why I imagine it as a U. You can imagine it as anything you want to. It is holding this tropomyosin in place with the active binding sites covered. When it already has loaded up with ATP, it's attached to the actin. It has one other site. That site is going to bind calcium. Calcium, remember, is going to be released into the cell. It's going to be collected in the cisterns. It's going to be released by way of the T tubules into the cell. It's going to interact with it. It's going to bind with the troponin. Troponin's going to undergo a configurational change, and then we're going to have the active binding sites exposed. 3 binds tropomyosin and actin. Calcium ions inhibitory substance that binds to actin. So as long as it's inhibitory, as long as it hasn't gone under configuration change, okay, it is keeping those sites, keeping tropomyosin in its place, and the active sites are not exposed. The tropomyosin, okay, spirals around actin to stiffen it. Like I say, you can think of anything, wire, duct tape, whatever you want to think of it. Physiology. This is what the intersection of a neuromuscular junction looks like. And obviously, we get nerves from the central nervous system, okay? It's going to come down to the muscle, all right? And so so we have an electrical signal, we have an electrical chemical signal coming down the nerve, okay? And that nerve is going to end up on this motor end plate, okay? And it's going to be, it actually doesn't touch it. There's a gap in between this, okay? There's a gap in between this. There's going to be a signal coming down the nerve. It's going to come to this gap, and then something's going to happen because we need to convert that. Now it was a electrical signal. Now it's going to come down to here and convert it to a chemical signal, and it's going to go across the muscle, and something's going to happen. So we're going to look at that in detail. This is actually what happens here. And we'll go through this. This action potential arrives at the terminal. So this is the nerve coming down. This is the axon end plate, okay? This is the muscle over here. This red stuff is muscle here. This is the junction, okay? This is the postsynaptic junction. This is presynaptic. This is called a synapse, okay? And so this is the space between the nerve and the muscle. These are specialized areas on the muscle which are receptive to these things that we're going to have released here. Okay, we're going to talk about them in just a second. So this action potential, that's what we call the signal in the nerve. It's an action potential. It's coming down the nerve. It arrives at the axon terminal, okay? When it arrives here, they are special gates here, okay? They are voltage gates. Well, this signal is an electrical signal, so it has voltage associated with it. It flips open these calcium voltage gates, and this calcium comes in. So this is not in the muscle yet. This is just here in the axon plates. This calcium comes in, okay? The calcium entry causes the synaptic vesicles. These are synaptic vesicles. So this is an enzyme. This is acetylcholine. Guess how it was made? We had a signal inside the nucleus, went outside the nucleus by way of RNA down to the rough endoplasmic reticulum, made this protein here. It was packaged in the Golgi apparatus and then placed into carriers, okay? Well, these carriers are going to come over here and fuse with the end membrane of the axon plate. So this calcium has caused some of these synaptic vessels to be released. and they contain acetylcholine, which is the neurotransmitter for skeletal muscle. There are several neurotransmitters. We're going to study maybe 3 or 4 in these 16 chapters, but this is the one that works on muscles, on skeletal muscles. Okay, so neurotransmitter, it diffuses. Remember, there's a space here. So it's got to go from here over to there. And so this, we're now in, this is intercellular. This is intercellular. So this is extracellular fluid. This is extracellular fluid. So it's going to diffuse across this extracellular fluid and bind to the receptors on the sarcolemma. Well, the sarcolemma is the plasma membrane. It's the plasma membrane of the muscle. Okay, so this is how the nerve signal gets to the muscle. The signal comes down to the nerve, and we'll learn about action potentials in chapter 11. So 2 chapters from now. We got 10 to go, and then 11. So the action potential comes down to the nerve. Okay, comes down to the axon plate, the motor end plate. There is a gap there. Okay, we're going to release a chemical called a neurotransmitter that's going to go across that gap. How's that neurotransmitter get released? When the signal comes down, which is an electrical signal, we have voltage associated calcium gates. When that voltage hits that calcium gate, that gate opens up, calcium comes in. Calcium then causes the release of the neurotransmitters, and the Golgi apparatus is going to fuse with the end of the neuron, its cell membrane, and that acetylcholine is going to diffuse across that cleft, across that space, and interact with the receptors. Remember, this area where we have this nerve muscle junction has this specialized area, usually in the center of the muscle fiber, because there's no reason for it not to be in the center, because all a muscle fiber does is contract, and it's going to diffuse across here. Okay, and so you're going to learn a lot about that as you go. When action potentials trigger release of acetylcholine, that's the abbreviation for acetylcholine, diffuses across the sarcolemma, which contain receptors for acetylcholine. Okay. Well, how do you give a substance that blocks transmission? So if you're going to have surgery and you don't want the patient to move around because you're going to do, you're going to do orthopedic surgery or something like that, and you're going to work with the big muscles and you want everything to be relaxed, then you're going to put a blocker in here. You're going to put a blocker in here. Okay. It's either going to take up the acetylcholine or block the receptors. All right. And so that is one of your muscle relaxers. Okay. You get a muscle relaxer and anesthesia, and that's what it does. It blocks the signal from from getting to the muscle cell and causing a contraction. So we've got this coming here. Acetylcholine is going to diffuse across. Okay. Membrane area on the muscle, generally in the center, that contains receptors for the acetylcholine, will depolarize. Okay. It's going to depolarize. In other words, the signal is going to be the signal that was coming down the nerve, converted to a chemical signal, going to cause the muscle to generate a signal and going to send that signal deep into the muscle cell. Okay. So one of the videos that you're going to watch, are 6 videos for the interactive physiology thing. You just have to watch those little animations. They're about 4 or 5 or 6 minutes, and there's some questions to answer. So you can watch them as many times as you want. You fill out the questions, you turn it in, you get full credit. It's an easy thing. One of the things it talks about, one of the videos is, and has questions about, is this excitation, contraction, coupling. Action potential travels across the sarcolemma and down the T-tubules. When it goes down into the T-tubules, okay, here are the cisterns, the 2 cisterns. Here is calcium stored in the cisterns. When that action potential occurs, this calcium is now dumped into the T-tubules. Remember, the T-tubules are going to go down into the cytoplasm, into the sarcoplasm of the muscle cell, and put calcium into the sarcoplasm. Why do we need calcium there? Because calcium is going to have to interact with troponin so that we can expose the heads on the actin so that the myosin fibers can attach to it. Calcium binds to troponin, and then we get a contraction here, okay? So we got the troponin here. Tropomyosin is blocking the active sites. We got actin here. We have calcium binding to troponin and removing of the blocking action. So when the calcium binds to the troponin undergoes a configurational change. When it no longer, I think of it, no longer holds the tropomyosin in place. And so all of a sudden those active sites are bound. Here's a myoglobulin head. This is myoglobulin, okay? This is myosin, this is myosin, this is myosin head. It is now going to bind to the active site. As long as the active site's covered up, there's no place for it to bind, okay? But now that the active site is open, okay, or available, it can bind to the active site of myosin, okay? It can bind to the active site of myosin. And so when it does that, there's going to be a contraction. Okay, we'll look at that in a little bit more detail. Muscle at rest, the I bands and the H bands are at their longest length. The sarcomere is its longest length. The calcium, and when the muscle cell is relaxed, is being stored in the sarcoplasmic reticulum. The troponin-tropomyosin complex covers the active sites. We have some ATP, stored ATP, but only a few seconds worth once contraction begins. So we've got to keep some ATP going. This just illustrates that same point that we looked about before. This is the illustration of several diagrams about our action potential coming down here, the acetylcholine release depolarization, going down through the entire length of the cell. The T-tubule, which is next to the cistern, is going to have calcium released, and we're going to get a contraction there. Sliding filament theory. This is a theory. Okay, this is a theory, not a law. Okay, it's not like Newton's universal law of gravitation. This is a theory as to how muscle contraction occurs. Okay, nerve impulses reach acetylcholine is released and goes over to the receptors. The sarcolemma depolarizes. T-tubules also depolarize, carrying action potential to the interior. Triggers calcium release from the cisterns into the intercellular space by way of the T-tubules. Calcium ions cause troponin to change shape and open the binding sites. Cross bridges form between the actin and the myosin. So we got cross bridges formed here between the actin and myosin. We had ATP here, okay? We have broken the ATP. The energy for ATP, adenosine triphosphate, is in that last phosphate bond, okay? We're going to need some energy here to make this sliding motion. We're going to need this energy here to make, so we have broken up ADP, that last phosphate bond. We have released the energy, so we now have energy. Cross bridges break as myosin pulls actin along the filament. So what happens is the myosin is pulling the actin. Okay, the myosin is pulling the actin. So it's got the energy of the ATP. The active site's exposed. It's going to attach there, and it's going to contract, and it's going to pull the actin. It's pulling the actin. It's pulling the actin centrally. Okay, pulling the actin centrally, so that the myosin stays in place. Okay, the myosin stays in place. The actin is going to slide over the top of the myosin. Now, this is not just one head. Okay. This is not just one head, because if it's one head and it released the actin, then the actin would snap back into its position. These are multiple myosin heads moving the filament in a concerted effort. Okay, multiple myosin heads trying to accomplish moving the filament. So the I band and the H zone disappear. Remember, the I band is the place where only actin is present, and the H zone is in the A band, and that's the place where there is no actin, there's only myosin. I never know how to take this back. So once the, how does it all stop? Once the signal from the nerve is stopped, okay, then the acetylcholine, a number of things happen, okay. It is either reabsorbed by the nerve end plate, okay, broken down and reutilized, or it can just diffuse away. Okay, remember, it is in this space here. It's in the space here, and it can be just diffused away. And there also is an enzyme for breaking it down. The calcium returns to the sarcoplasmic reticulum by active transport and can be stored there, bound to the calcitrin, to this protein. It's going to be bound to that. And the troponin and tropomyosin move back to cover the active sites. ATP is resynthesized to cover the myosin heads. The filament slides back to the resting position. In rigor mortis, if no ATP, rigor mortis, after somebody dies, the body gets stiff. If there's no ATP can be produced, then the cross bridges cannot unattach, because we have no ATP to prime that head again, okay? We have no ATP. If somebody's dead, they're not doing cellular metabolism and cellular respiration, so they're not forming ATP, run out of ATP, and then the body goes into rigor mortis, but eventually the proteins start breaking down, and so these attachments are released, okay? And that's as far as we're going to talk today, okay? That's as far as we're going to talk today, because that's about halfway, because we're going to talk next week about the motor end units. We're going to